First of all, I want to thank the organizers for giving me this opportunity. I think, though, they made a mix-up and they put me in the session because I am not one of the wise elders. I am a graduate student in Rachel Whitaker's lab, and I work with archaeal viruses, and sometimes I feel it's like 100 years back when phage people were starting off, because there's really not a lot known about these viruses. So I'm going to just show you some new data that we think it's exciting that we found, and I'm just going to share it with you. I don't have any funny anecdotes either, so I am sorry. Okay. So, with the aid of all molecular tools, we found that viruses are everywhere. Viral diversity is amazing, and many of you here are responsible for all these findings. Finding viruses basically infect all domains of life, and probably every single organism on Earth is infected by viruses. And so, there's still a lot to be uncovered in regards to viral diversity, but not only viral diversity, but also how the interactions with their hosts shape biological communities, not only microbial, but in general, biological communities, and this also remains to be uncovered. And so, how do viruses shape these communities? There's still a lot we don't understand. People have thought about this many times, thinking about it as this predator-prey dynamics, but it really is much more than that. And until we understand how these interactions shape populations and communities, we can't really model them, because people do a lot of modeling based on parameters that are based upon few model organisms in the laboratory, but really there's a lot of things that are missing from these models. So what's missing? This is just a few examples. So from the virus side, there's other types of virus lifestyles. So when we think about phage, we think about the lytic and the temperate phage, right? But there's other types of lifestyles, and especially in archaeal viruses, there's a lot of persistent chronic infections. So these cells are infected and they're constantly producing virus. Not because of a switch that they go lytic, they're just shedding virus all the time. Also, there's proviruses that are carrying advantageous genes, and that's important too. It's very important, actually. So that also has to be taken into account. But also, we need to think about other types of host lifestyles. So, for example, the recently described CRISPR adaptive immune system. So usually, when there's been modeling, people consider cells to be susceptible or resistant. Many times, this resistance are surface receptors that have trade-offs. And these types of models, having a trade-off allows coexistence of susceptible or resistant cells. However, if you look at immunity, immunity is very different for the cell and what it entails for the cell. So it has to be a separate category than resistance. And, well, another type of response from the host to virus is dormancy. And so we studied these interactions in acidic hot springs. They're great environments to study host-virus interactions. Why? Because they're low-complexity environments. They're geographically isolated. There's a limited number of microbial hosts, and viruses are the only known predators. So that's great. And so in the system, we focus on the species Opholobus. Icelandicus mostly in our lab. This is a hyperthermophilic Cranarchaean, as Ken said a few talks ago. And it lives at a pH of 80 degrees and a temperature of 80 degrees and a pH of 3.5. And also, we know a little bit about this organism because it has been the model archaea to study hyperthermophilic Cranarchaea, so we know a little bit about its cell biology, biochemistry, and genetics. Not a lot, though. And also the Sulfolobus viruses that, if you were paying attention to Ken's talk, are absolutely unbelievable. They have amazing morphologies. They form pyramids to exit the cells. How cool is that? I mean, honestly. I think now all of you are going to go and work with archaeal viruses. And these viruses have been isolated in hot springs all around the world. They were originally discovered by Wolfram Zillig, and now There's plenty of groups isolating viruses. Those are pictures of viruses we have isolated in our lab. And I will focus on one particular virus, the Sulfolobus spindle-shaped viruses. These guys have a circular double-stranded genome that range between 15 and 24 kilobase pairs. They like to clump. So this is a picture I took of one virus that my undergrad isolated, and they have sticky fibers, so they usually tend to cluster together. And these viruses also are found in the genomes of Sulfolobus. So in our laboratory, there's a lot of sequenced genomes of Sulfolobus, and many of them have these integrated proviruses. They integrate into the tRNAs. These viruses, again, are found in hot springs all over the world, and they have, they're in isolated populations. They have a biogeographic structure. And so and where's the pointer? Does this have a pointer? It's on the top. Yeah. And so this is looking at the core genome of all the SSVs that have been found either as particles or proviruses. Another cool thing about Sulfolobus and viruses is that all of the Sulfolobus genomes sequenced so far have this CRISPR-Cas adaptive immune system. So a brief overview of this adaptive immune system is that, upon infection with a virus or a mobile genetic element here in the host has this CRISPR locus, which consists of the repeat spacer array and the Cas or CRISPR-associated genes. A part of this virus is integrated generally into the leader end of this repeat spacer array. This is transcribed and processed into small CRISPR RNAs that act as sentinels. So upon another infection with the same virus or a very related virus, this little CRISPR RNA will recognize and recruit all of the machinery to degrade this invading genetic element. And so when we look at these genomes of Sulfolobus in nature, we see that they have these spacers matching these SSV viruses. And so we are focused in two populations, in Kamchatka, in Russia, and in North America. And if you look, the spacers that match SSVs are very different in both populations. But it doesn't mean that both viruses are not present in both populations. Actually, they're quite abundant in Yellowstone. So we don't know why they're different or why these viruses have evolved differently and how this affects colonization dynamics and population structure. So we really want to tackle that question. And so we selected one particular pair to look at. So this was RJW002, which was isolated from Kamchatka, and SSV9, which was formerly called SSVK1, and Ken was part of the group that isolated this virus, and he kindly provided it to us. We selected this pair because this RJW002 has a perfect CRISPR match against this virus. And so we expected to see what other people had seen with SSV1, which is the model SSV virus. The cells get infected, they slow their growth a little bit, and then they just keep growing, and they keep shedding virus. As Ken mentioned, some of these SSVs are triggered to produce when they're induced by any stressor like UV, but some others lack this transcript, the UV-inducible transcript, so they're constantly shedding virus. And so we challenged these cultures with a very low amount of virus, and what we saw was that even at that low multiplicity of infection, it affected a large population of host cells. So we see that there's a lag of growth in here in the OD. You can see there's a lag for 24, sometimes it's 48 hours, and when we did viable counts, the cells weren't really growing on this first 24 hours. And so we were curious as why that very few viruses were producing such a big effect. And so another student in the lab measured the absorption constant of the virus, and it ends up it's low, which suggests two possible scenarios. Either absorption is reversible, and so the virus is touching the cells but being released, and so basically even it's a low number of viruses, it's touching many different cells, or sulfolobus have very, very few receptors, and when the cells take in the virus, the cells are producing something that is making the cells go into some sort of arrest or dormancy. And so when we looked actually at these cultures, we did thin sections of the cultures over time. We saw that there was a sharp increase in these cells that look rounded and empty, which we call dormant. And these cells peak at 24 hours. Most of the cells look like this, and then they start dropping off as the cultures also recover their growth. And so why do the cultures recover after this lag? And so our hypothesis was that it was because of the CRISPRs, that the CRISPRs were eliminating the virus from the culture. And so we constructed a set of in-frame deletion mutants that deleted parts of the CRISPR system we thought would have no effect, like the Cas1 protein, which is important in acquisition, but since this cell already had a spacer, we didn't think it would have an effect. And we also deleted the A2 array, which is another array which doesn't have a match to SSV9. And we made also 3 other deletion mutants. One of the A1 repeat spacer array, which is the one that actually has the match to SSV9, of Cas3, which is the interference protein, the one that chops up the invading nucleic acids, and of Cas6, which is the one that does the processing of the CRISPR RNA. And we hypothesized that those would abolish immunity. And so they did. So what we found was what we expected. These guys, the wild type or the RJW002, the deletion of Cas1 and A2, were still immune, so no viral particles were produced in those cultures. And the DNA went down. And then we have the immune deficient strains where the virus was able to replicate, and so we find plaque-forming units or viral particles in the supernatant, and the viral DNA replicates. So then when we looked at those immune deficient strains, these guys show a very different trajectory than those with immunity, with no significant increase in OD. So the other ones, even though the virus was not replicating and the amount of virus present was very low, they have this lag, but then they recover. The ones where the virus is replicating don't recover throughout the course of the experiment. So when we looked at these cultures, at the susceptible ones that are producing virus, we see the same rounded, empty phenotype over time, and it increases again at 24 hours, but it stays high because these cells don't recover, so it's like a prolonged state of dormancy. And so we wondered if this death that we see in these cultures, because when we do viable cell counts, more than 99% of the cells lose viability by 24 hours. We wonder if it's this prolonged dormancy that ends up leading to death. One thing I want to note is even in cells that were unviable, lysis was never observed. We never saw broken cells. And so to test this, we took inactivated virus. So we UV irradiated the virus and added it to the cultures and saw if that had any effect. So in these cultures, the virus is not replicating, it's not active. However, it shows the same phenotype than in the RJW02, where the cultures lag and then recover. But then we decided, okay, so if in immune deficient cultures, what's killing the cells is the presence of the virus, keeping these cells dormant until they die, what if we keep adding the signal, this UV inactivated virus, and see what happens? So we took the cultures and added 3 doses of UV inactivated SSV9. And what we see is something similar to what we observed with the immune deficient strains that were producing virus. They don't really recover. And when we look at cultures that have been incubated for 72 hours, there's really no growth at all to be detected. So that's what I wanted to show you. That this viral-induced host cell dormancy can lead to cell death with no viral replication of lysis. There's other systems like TA systems or ABI systems, but they require the virus to go into the cells, and actually the virus starts replicating and they target these different steps. This is just by coming into contact with the virus. And a very small amount of virus can have a very big impact on the population. So that's something we really need to take into account when we're doing these models, they're doing this modeling, because sometimes we see a lot of these dormant cells and we don't necessarily understand why, because the viral abundance, like in the environment, is low, but it doesn't really take a lot of viruses to cause a big impact on the population. And with that, I'd like to thank my advisor, Rachel Whitaker, and the members of the lab, especially Dr. Qingyi Zhang, who created the mutants and developed a genetic system for us to start really asking these questions. Before him, we were basically like 60 years ago in molecular biology, we couldn't even make mutants. So, we're starting to be able to ask new questions. I also wanted to point out that Elizabeth Rowland has a poster, and she has been looking at these really cool resistant mutants that are resistant to virus independent of CRISPR, and also they somehow are resistant to this dormancy. So it's cool if you stop by and look at our poster. Our collaborators, Dr. Mark Young at Montana State, Dr. Joshua Weitz, and NSF for funding. And with that, I would take any questions. Thank you.